Hello students, today we are going to study a very important topic of the cell biology, and that is known as the cell membrane. The name cell membrane was first of all given by the two scientists known as the Nageli and the Kramer, and this cell membrane is also called as the plasma lemma, and this plasma lemma name was given by a scientist known as the Plover. Cell membrane is a common membrane. It is found around the plant cell as well as the animal cell. If we want to study the cell membrane, we have to study the structure and the function of the cell membrane. To study the structure of the cell membrane, we have to study the three models which explains the structure of the cell membrane in a better way. The first model was known as the sandwich model. The sandwich model was given by two scientists known as the Danieli and the Davison. This model is also called as the trilamellar structure. Now the question arises that what is a trilamellar structure? The trilamellar structure suggests that the plasma membrane is made up of altogether three layers. There are two protein layers on the either side, and sandwiched in between the two protein layer is the bimolecular layer of the phospholipid, and that's why we can say that the structure of the plasma membrane is trilamellar. You can see in the diagram I have made the two protein layers on the either side. This is one protein layer having the thickness of the 20 angstrom. This is the another protein layer which is having the thickness of the 20 angstrom, and in between these two layers there is a bimolecular layer of the phospholipid which is having the thickness of the 35 angstrom. So we can say that the entire thickness of the cell membrane is roughly around 75 angstrom. Now the bimolecular layer of the phospholipid is having the head and the tail, and we can also say because of the presence of the head and the tail in the bimolecular layer of the phospholipid, we can also say that it is amphiphatic. It is amphiphatic in nature. What is amphiphatic? Amphiphatic means the bimolecular layer of the phospholipids are having the head and the tail. I am marking here, these are the heads of the phospholipid, and these structures are the tail which are facing towards each other. These are the tails of the phospholipid. Now these heads are polar. These heads are polar and they are hydrophilic. Now what is the meaning of the word hydrophilic? Hydrophilic means that they are water loving, and these heads are associated with the protein layer with the help of certain bonds, and these bonds are called as the ionic and the hydrogen bonds, while these tails are always non-polar. These tails are always non-polar, and the tail is always hydrophobic. Hydrophobic means they are water heating, and these tails are associated with each other. These tails are associated with each other with the help of special forces, and these forces are called as the van der Waals forces. This entire structure, that is the trilamellar structure, can also be represented as PLLP arrangement, means the two protein layers on the either side, and sandwiched in between this protein layer is present the bimolecular layer of the phospholipid. But nowadays this sandwich model is not accepted. Now the question arises that why it is not accepted? Because the protein arrangement what was shown by the Danieli and the Davison in this sandwich model is incorrect. In the later models you will see that the protein arrangement is little bit different than what was shown by Danieli and Davison in the sandwich model. So this was the first model and was named as the sandwich model. Now coming to the next model, and that model is called as the unit membrane concept given by a scientist known as the Robertson. This Robertson also says that the structure of the plasma membrane is trilamellar. He also accepts that the plasma membrane is trilamellar, means the plasma membrane is made up of two protein layers and a bimolecular layer of the phospholipid. But what was the difference between this model and the sandwich model? So you can see that he says that he gave the concept of the unit membrane. What he says, that not only the cell membranes but membranes surrounding the cell organelle is also trilamellar. What he says, that the membrane which is surrounding the cell is trilamellar definitely, but all the membranes found in the cell are also having the same structure. Means what he want to say, all the organelles, whether it is mitochondria or the Golgi body or the endoplasmic reticulum, are surrounded by the same membranes. Means nature of all the membranes found in the cell is similar, and therefore for all the membranes present in the cell, he gave the concept of the unit membrane. But this model was also not accepted. Robertson model was not accepted. Why this model was not accepted? This model was not accepted because of the two reasons. Because this model was not able to explain the elasticity of the cell membrane, as well as it was not able to explain the selective permeability of the cell membrane. Therefore, this model is also discarded nowadays and it is also not accepted. But to explain the structure of the cell membrane in a better way, the best model which is available at present to us is the fluid mosaic model, which I have presented in front of you. And this fluid mosaic model was first of all given by the two scientists known as the Singer and the Nicholson. What is this model basically? This model says that plasma membrane is semi-fluid in nature. What I am saying, that plasma membrane is semi-fluid in nature, and in this semi-fluid, the lipid, means I want to say the phospholipid, and the intrinsic proteins form a mosaic pattern. They form a mosaic pattern. Now, what is this? That what do you mean by mosaic? Means what this model says, in simple aspects we can say that the phospholipid component of the cell membrane is providing the fluidity or the liquidity to the membrane. So what is the liquid part? Who forms the liquid part or the fluid part of the plasma membrane? Fluid part is actually formed by the phospholipid, and phospholipids are basically what they are unsaturated fatty acids. But what about the proteins? What are the proteins? So proteins are present in the form of icebergs in sea of lipids. Why we have used the word sea of lipids? Because lipids are providing the fluidity or the liquidity to the cell membrane, and in the sea of the lipids, the proteins are present in the form of icebergs. So protein icebergs in sea of lipids. The arrangement is just like that type. In the sea of the lipids, the proteins are present in the form of icebergs. Okay. Now, to explain this model in a far better manner, we can also take an example of a utensil. Which is actually having the gulab jamuns. You all know about the gulab jamuns. That is a sweet dish used in the India. So these are what the gulab jamuns. These are the gulab jamun, and this is what the liquid. As you know very well that gulab jamuns are dipped in a liquid, right? So what is the arrangement? The arrangement of the phospholipids and the proteins in the cell membrane is exactly like this type. That is, we can say that like the gulab jamuns are dipped in the sugar in a utensil, exactly in that manner only the gulab jamuns are dipped in the liquid. So we can say that we can treat the gulab jamuns as the proteins, and we can treat the sugar solution as the phospholipid. Are you getting what is the arrangement of the phospholipids and the proteins? The arrangement of the proteins and the phospholipids is exactly same, just like the gulab jamuns are dipped in the sugar solution in a utensil. Here you can see that gulab jamuns can be treated as proteins and the sugar solution can be treated as phospholipids. So entirely we can say that plasma membrane is semi-fluid in nature, and the lipids and the intrinsic proteins are making a mosaic pattern. Here the phospholipid is providing the fluidity to the membrane, while in the sea of the phospholipids the proteins are actually embedded at the different places. Just like the gulab jamuns are dipped in the sugar solution, exactly like that types, proteins are dipped in the sea of the lipids. This is the best model which explains the structure of the cell membrane in a better manner. You can see here that the arrangement of the proteins, you can see here the arrangement of the proteins which was shown by the Singer and the Nicholson is exactly different from what it was shown by the Danieli and the Davison. Danieli and Davison said that the protein layer is covering the phospholipid entirely, but no, they say that the arrangement of the protein is little bit different here. You can see in the diagram that there are two type of the proteins. There are two type of the proteins. One are known as extrinsic or peripheral proteins. Now what are these extrinsic proteins? They are present on periphery. They are present on periphery and can be removed easily. These are the peripheral proteins. You can see here that I have made certain proteins at the surface. They are not embedded inside the cell membrane. They are present at the periphery, and therefore they are called as the extrinsic or the peripheral proteins. The next category of the proteins are called as intrinsic proteins. Intrinsic proteins can also be called it as integral proteins, and they cannot be removed easily. Means they are stable proteins and they cannot be removed easily. You can see that these are the intrinsic proteins, and there are few more proteins and they are called as tunnel proteins. There are few more proteins which are known as tunnel proteins and which are present from one end to the another end. You can see that these are presents from the one end to the another end. Are you getting? You are able to see the three proteins. One is present at the periphery, known as the peripheral protein. One is present at the center, that is inside the cell membrane, and are called as the intrinsic protein. And there are few proteins which are actually meant for the transport of the substances, and they are present from one end of the plasma membrane to the another end of the plasma membrane, and these proteins are called as tunnel proteins. Say for I have given an example here. The best example of the tunnel protein is the porine protein. Okay, so this is the arrangement of the protein. So these three type of the proteins are arranged in the bimolecular layer of the phospholipid. So this is the best model available at present date, which explains the structure of the cell membrane in a proper manner. But before ending the fluid mosaic model, we must also know about a term, and that term is called as flip-flop movement. Many times question is asked from this flip-flop movement, that what is this flip-flop movement? Flip-flop movement is not allowed here. Flip-flop movement is not allowed here. Means what? The intrinsic proteins cannot move from one lipid layer to another layer. Are you getting? Say for if this is the intrinsic protein, so the movement of the intrinsic protein from one lipid layer to another lipid layer is not possible. It is not allowed here. Flip-flop movement is not allowed here. Means the intrinsic protein cannot move from one lipid layer to the another lipid layer. This movement, this flip-flop movement is not allowed. So always remember that flip-flop movement of the proteins, that is the movement of the proteins from one layer to the another layer, is not at all allowed. So these were the three models to explain the structure of the cell membrane. Okay, now we must know about the function of the cell membrane also, that what are the functions of the cell membrane. We must know that the chief function of the cell membrane is that the cell membrane is important for transport of substances. The chief function of the cell membrane is transport. Now what type of the transport? So the very first mode of the transport is active transport. First mode of transport is active transport. Here chemical energy required. Here chemical energy required for transport. Why? Because the substances are moving from low concentration to high concentration, and whenever the substance is moving from low concentration to high concentration, certain amount of energy is required. Means what? The substances are moving against diffusion gradient. Means diffusion always occurs from high concentration to low concentration, but they are moving against the concentration gradient. Means the substances are moving from low concentration to high concentration, and therefore some chemical energy is required, and such type of the transport across the cell membrane will be called as what? The active transport. The second transport is known as the passive one, passive transport. Now what does this passive transport means? Passive transport do not require energy. Why? Because the substances are moving from high concentration to low concentration, and here substances are moving along concentration gradient. Means here the substances are moving from high to low, and therefore they are not moving against the concentration gradient, they are moving in favor of the diffusion gradient, and hence the energy is not required. So passive transport, one was known as the active transport. Now the third one, that also comes under the transport only, and that is the third category, is exocytosis. Exocytosis means exit of substances from cell. Certain substances are thrown out of the cell by a process called as exocytosis. And the fourth type of the transport is endocytosis. What does the endocytosis means? Entry of substances. Now the entry of the substances inside the cell can occur by two methods. One is known as pinocytosis and the another one is known as phagocytosis. Phagocytosis means what? Engulfment of solid particles, and this is intake of fluid substances. Because here the intake of fluid substances occur, it is also called as cell drinking. We can also use the term cell drinking in place of the pinocytosis, while in the phagocytosis we can use the term cell eating. So this was the main function of the cell membrane, and that was known as what? The transport. This was the function number 1, known as transport. But if we talk about the other functions of the cell membrane, so we must know that it is helpful in the exchange of gases. It is helpful in exchange of gases. Not only this, the cell membrane is selectively permeable, means it allows certain selective particles to pass through it. Not all the substances can be passed through the cell membrane. And the fourth point, which is very, very important one, and that is known as cell to cell recognition. Very, very important point. Cell to cell recognition means what? Cell to cell recognition means that the glycoprotein and glycolipid content of the cell membrane are responsible for cell to cell recognition. The acceptance of any organ or rejection of any organ during the organ transplantation is just because of the glycoprotein and the glycolipid. Means cell to cell recognition means what? Say for we cannot transfer the liver cells into the heart or the heart cells into the liver. Why? Because the cells are having the power of cell to cell recognition because of the two contents of the cell membrane, glycoproteins and the glycolipids. They can reject also, they can accept also. So we cannot transfer the different cells in different organs. Liver cells cannot be transferred in the kidney, kidney cells cannot be transferred into the liver. So this was the cell to cell recognition. So dear student, this was the function as well as the structure of the cell membrane. Okay, so you must remember the structure also, and you must remember about the function also. Okay, so this was the description of the cell membrane. If you have liked this video, subscribe my channel and also like and share this video. Thanks a lot. We will be back soon. Thank you.